Hello, and welcome to the Red Bank Humanists podcast. Each month, Red Bank Humanists hosts a public forum featuring speakers and topics covering a wide range of subjects of special interest to the humanist community. To learn more, including information about our monthly book club and our Drinking Post-Theologically events, visit redbankhumanists.org and follow us on Twitter at Red Bank Humanists.
Promoted by the International Darwin Day Foundation, Darwin Day is an international celebration of science and humanity held on or around February 12th, the day that Charles Darwin was born in 1809. Specifically, it celebrates the discoveries and life of Charles Darwin, the man who first described biological evolution via natural selection with scientific rigor. More generally, Darwin Day expresses gratitude for the enormous benefits that scientific knowledge, acquired through human curiosity and ingenuity, has contributed to the advancement of humanity. Recognizing science as an international language accessible to all individuals and societies, the International Darwin Day Foundation provides a new global holiday that transcends separate nationalities and cultures. In Darwin Day, we are able to recognize the diversity among us while celebrating our common humanity and the universal understanding we all share. The Red Bank Humanists has been celebrating Darwin Day at our February forums for many years. It's our favorite holiday, and we look forward to it every year. And here to help us to celebrate today is our special guest speaker, Michael Palladino, Dean of the School of Science and Associate Professor of Biology at Monmouth University, right down the road in West Long Branch. Mr. Palladino specializes in male reproductive biology and cell and molecular biology. He received his BS in biology from Trenton State College. He studied nucleic acid biochemistry and molecular biology of DNA triple helix formation at Princeton University. Earned his PhD in anatomy and cell biology from the University of Virginia. He was a faculty member in the biology department at Brookdale Community College. In 1999, he joined the biology faculty at Monmouth University and was appointed dean in 2008. Michael Palladino has received many awards for his teachings and research. He is an advocate for students and research and education, and he's the author of Understanding the Human Genome Project. We are honored to have him here for our Darwin Day celebration to give his presentation on designer babies, what Darwin couldn't predict. Won't you please give a warm welcome to Michael Palladino.
Thank you for that obituary, Stephen. I appreciate it. Sincerely, Stephen, thank you, and Judy, thank you very much for the opportunity to come and speak to you this morning, and to my colleague Stan Green over at Monmouth University. I appreciate the invitation. What I hope to be able to do for you in the time that we have, and maybe as we get rolling, we can turn the lights down a little bit so you can see this better, is I hope to really have a conversation with you this morning, a discussion really, and I hope to leave you with more questions than answers, because part of what we're going to talk about today involves what is a relatively small time scale on Darwinian evolution, and the answers to some of the questions I'm going to pose won't be known still for quite some time. So I hope to leave you with some provocative things to think about. Let's have some questions as we go along if that works well for you, and then if we find that we're getting too close up against the end of time, so I think we can go till a little bit before 12 or so, as I understand it, and maybe we can continue to take questions at the end. And I'd be happy to hang around for a little bit and continue speaking to you. And if you see something that you particularly like in the slide sets as we talk about this, I'd be happy to leave a copy with Trudy, and she can share that with you if you're interested in following up a little bit. Okay, so you heard a little bit about me. Let me tell you one or two other things to sort of set the context for what we're going to be talking about today. In terms of the kind of research that I do and my research interests, I'm what you'd call a molecular reproductive biologist. Okay, so what does that mean? So as a molecular biologist, I clone genes and study genes, and we look at what those genes do. And when you say you're a reproductive biologist, that usually makes for very interesting cocktail party conversations, because when you tell people you're a reproductive biologist, they usually assume that you're some sort of sex therapist, Dr. Ruth type, and they start to tell you a lot of interesting, intimate details that you really don't want to know too much about. But when you tell them what you really work on, then usually they sort of glaze over and they're not too interested, and then the conversation changes very quickly. So it could be the killer at the cocktail party too. This is what I really work on. In the biggest sense, we work on what we call protection of the male reproductive tract. I look at antimicrobial properties. In other words, how does the reproductive tract know when bacteria, viruses, and yeast invade the tract, sense that those bacteria, viruses, and yeast are there, and then elicit mechanisms at the molecular level, genes and proteins, to be able to clear those bacteria and viruses. And this is a pretty big deal in the bigger sense because we know that bacterial, viral, and yeast infections of the male reproductive tract are a significant cause of subfertility and infertility. And the same thing happens on the female side as well. We also look at how changes in oxygen levels in the reproductive tract affect cell development, particularly developing sperm cells. There are a number of different clinical conditions in which oxygen levels change, and that affects the reproductive tract. So again, we study things at the cell and molecular level with respect to how does the reproductive tract know there are changes in oxygen levels, and what kinds of things can the reproductive tract do to try to fix and protect those cells from damage. So a lot of what I'm going to talk about today in this designer baby context is coming at this from a male fertility side of things. And I want you to keep in mind, too, that there are significant issues from the female side that we can layer on top of this, and that would be a conversation for really another day. Okay, so you've likely seen this photo before, perhaps. How many folks are relatively familiar with Louise? Okay, test tube baby. When was this? What year? 78, I heard. Excellent. 1978. Okay, baby Louise, the world's first test tube baby. You're going to hear me talk a lot today about the concept of IVF, and IVF in the context of what we call assisted reproductive technologies. Okay, and I'll show you another type of assisted reproductive technology today, but IVF was really one of the first. And I'll make some assumptions about IVF, and maybe they're not true, and you'll help me if these are things you're not familiar with. But the basic concept of in vitro fertilization is that you can harvest from the female one or typically more eggs, and there's a variety of ways you can stimulate a female to produce eggs hormonally, but you can harvest those eggs, you can maintain them in a laboratory dish, and you can take developing sperm from a male and get the sperm and the egg to fertilize in vitro, right, in a glass dish, in a test tube, if you would. You let the embryos grow to a certain period of time, and then you implant one or typically a few more embryos. So you may implant as many as 3 or 4 with the hopes that one embryo takes. And sometimes, of course, multiple embryos take, and that's in part one of the challenges that happens with in vitro fertilization, okay? And then that embryo then has to go through roughly a 9-month gestational process in utero, right? All right, so IVF starts in the UK, and someone out here in the audience said in 1978, and folks in the United States were struggling with this for a while. It took, in the UK example, it took about 15,000 attempts at IVF just to get a sperm and egg cell to actually do what you wanted them to do in a tube. And so a couple years later, the US scientists who developed this had figured it out a little bit better. It took them significantly fewer attempts. And lo and behold, we were on our way to in vitro fertilization as really the first true assisted reproductive technology, anyway, at least for humans. And in vitro fertilization, by the way, had actually been going on for quite a long time before that on the animal side, and there's all sorts of interesting issues with respect to animal evolution that we know a little bit about already based on how IVF has worked. So when IVF first got started in the UK, there were a lot of concerns. You had the Catholic Church generally being very concerned about in vitro fertilization and what it meant. You had at that time about 5,000 people almost immediately sign up for IVF, even though people really didn't even know what the costs were. The costs were actually pretty high, and they still continue to be relatively high. Do you have a sense for how many babies since that 1978 watermark have been born by IVF? Venture a guess. Yeah. I'm sorry, repeat that. So 200,000, but I don't know that they keep real statistics. That second part in particular is really something I want to drive home today. Do I hear another number? Okay, at least 250 or more. I'm taking bids. 500,000. 500,000. I'm still taking bids. Over 1 million. 10 million. Okay, I see where we're going here. Okay. 4 million babies worldwide. Okay, 4 million babies worldwide. The latest best guess numbers, and I say best guess, they're probably an underestimate, probably underreported. Okay, so a technology that was thought to impact relatively few children early on, okay, now is out to 4 million or so in size. And this is just one assisted reproductive technology. Okay. So a number of concerns popped up around that time with IVF. You may not be able to see my particularly bad cartoon here. And it says they couldn't tell me anything about my genetic father apart from the fact that he'd been a frequent donor. Okay. And this is, and I don't mean to say that to make light of the issues we're going to talk about today. Most of what I'll talk about is, I think, fairly serious today. But a number of concerns were raised immediately about in vitro fertilization. I want to explore some of those a little bit. And one of the things you're going to hear me talk about a good bit today is that immediately there were concerns about regulating this industry. And I can tell you that as an industry, IVF is one of the least regulated industries that's out there, and that continues to be a concern. I'll give you some examples of that. And there's lots of different aspects of what regulation could involve here. Right up front, people were very worried about this concept of unnatural conception. Okay, it's not natural. My reproductive bio colleagues who work in IVF say, you know, it's not nearly as fun as natural conception, but it can work. Okay, long-term health effects, of which there can be many, and that's something I want to talk a bit about today, and we can start to explore that a little bit. But keep in mind, on the evolutionary time scale, we're still talking about a relatively short period of time since IVF started, okay? And we're really only starting to look at second-generation IVF offspring in a serious way, okay? So for time scale here, it's relatively short. Significant concerns about the likelihood of inbreeding, okay? Sounds like a pretty far-fetched thing. I'm going to show you when we start to look at some numbers, especially in very small, geographically isolated countries, that likelihood can go up reasonably substantially. Okay. And then, of course, genetic risks associated with that, potential inbreeding, but other types of IVF and assisted reproductive technology concerns. And I should have probably said at the outset, as we're going through this, I don't want to push the panic button for people with respect to IVF, whether you've gone through IVF or are contemplating IVF. But what I want you to be able to hear is a realistic view from what many in the scientific community are very concerned about with respect to these approaches. Storage or disposal of unused embryos has really only gotten a lot of attention relatively recently, but it's been going on for a long time. So when I mentioned IVF before and I said that you have multiple embryos and you only implant a few, the remaining embryos are typically frozen away or they may be discarded. And some of those embryos, of course, have been the source for embryonic stem cells, which have gotten a lot of attention these days. But there are certainly ethical issues around what one does with the storage or disposal of those unused embryos. Cloning. Early on, newspapers were filled with images of the so-called test tube baby and these concepts that we'd start cloning offspring. Okay, and that's not really the case at all. I can tell you that of all the people I know who work in this area and who work in stem cells, I don't know any good scientist who is interested in human organism cloning, okay, in terms of producing offspring. Was significant amount of concern about using in vitro fertilization from what are so-called non-traditional couples, okay, whether that was older couples or lesbians or gays in various ways, okay, and these were a lot of literature and sort of a lot of discussion about that. And more recently, in particular, in several countries which we'll talk about here, have really been grappling with this issue of should you limit the number of children per sperm donor? Should you even limit the number of times someone can donate? And then should you limit the number of children that can be spawned by that donor? Now, I don't mean that this is an all-inclusive list of the concerns that popped up in 1978 and the years since then, but it gives you a bit of a landscape of a lot of the things that people were worried about. But early on in here, okay, left one other one for you here. Early on in here, biological fathering, okay, and the concept of someone being a biological father as a result of a sperm donor was a pretty significant discussion point, because what would that mean? And that led to some issues in terms of whether people should even know who their biological father, and hence my cartoon up here. But early on, the idea of selection, of using in vitro fertilization and the concepts of selection really weren't raised, in part because we didn't really have the technologies to really do true selection, okay? The selection being that you can allow a couple who may be infertile for either a male or female factor. They were infertile, they can have children, okay? But this concept of then selecting that embryo even further wasn't really well thought of, okay? And what I'm going to show you is that the concept of gender selection really became one of the first aspects of selection through in vitro fertilization. So if you're with me so far, we already have a level of selection. We've enabled people who may not be able to have children naturally, okay? And you've now enabled them to do that. And now you have the ability to select even further the particular embryos that one is using. And so people who started worrying about this very early on started to contemplate, started to propose what some of the implications might be. And this notion of what the impact on the human gene pool might be was really pretty much an unknown, okay, especially in very geographically isolated areas. And most people disregarded comments like this at the time because they said, well, this will only be a technology for the rich. You'll only get a few thousand people, and now you see some 4 million plus, and I've only shown you one assisted reproductive technology, okay? Well, with the economy these days, one can make a little bit of money. You may not be able to see the signs of sperm donors needed. These pop up all over college campuses all the time, around pubs and brewing houses and other places, including my home academic institution just down the road, Monmouth University. Because groups actively go out and solicit sperm donations from men, okay? Particularly college-age men, because they're presumed to be educated. They may come from families with a little bit of money, and they're considered to be relatively healthy. Okay, so there's a sort of a target audience there for sperm donors. With the economy these days, sperm bank donations are picking up. Okay, and it's not a joke. Okay, it's a particular reality. And if you look at articles like this is a little bit of a joke. You wouldn't make a great living doing this, okay, part-time, but at $150 or so a donation or more, I can tell you in two of the clinics where I did a fair amount of training, we'd see donors come in to provide sperm samples, and we knew if it was Tuesday morning, so-and-so was coming in before his 10 o'clock psychology class, okay? And you'd see people come in fairly regularly throughout that week. And on the male donation side, there are restrictions to how frequently someone can donate, largely because they need to have sufficient quantities of sperm to be able to produce. But it doesn't mean they won't try or change their name or do all kinds of interesting things, okay, at $150 a donation. And what's happening in other countries where this is a much more regulated industry, such as the UK and Australia, if you can see this here, they're seeing substantial shortages, in large part because part of the regulation that those countries have put into place is legislation now that gives children the right to go out and find out who their donor parent was. Okay, and we don't have that in the US, which is somewhat symptomatic of a series of regulatory issues in the US that we want to talk about. So we have countries that were sort of out and ahead of this and dealing with some of the regulatory issues, good or bad, okay, whereas here we are largely dealing with, generally speaking, a largely unregulated industry, okay. This issue of the number of offspring one can spawn is a pretty significant concern, okay. You may have seen articles like this. I know we're having some shifting images here, and the computers should be down here. One donor, one sperm donor, 150 offspring. This particular individual got a lot of attention for having fathered at least 150 offspring. I want you to consider the implications of that, 150 offspring, okay. Now, we know that in the U.S. and elsewhere that IVF-based conception practices and some other practices I'll show you in a moment are becoming more common, but as you heard from the outset, we don't have a great sense entirely of the numbers of individuals born to IVF, nor do we track them particularly well. Okay, but we know as practice, in vitro fertilization practices are becoming much, much more frequent than they were. Okay, so we don't have a good sense of this number. Okay, because we don't, as a country, really look to collect that data systematically from IVF clinics. Okay, so we have reasonable data, but not great data. We know from groups like the American Society for Reproductive Medicine, when they query donors involved in IVF, we know a little bit about their preferences. So this was a pretty large-scale cohort of individuals who use IVF, okay? And of those individuals, you can see 38% of them prefer having anonymous donors on the sperm side, and about 20% would like to know, and then about 42% say it doesn't really matter to them. They're looking to have a child. Okay, well, part of the reason why I mention this, yep, we have a question here? Good question. So the people who were being born were not asked here. These are the parents, so to speak, in the sense that the person who comes in to request a sperm donation, so these are females, okay, coming in to request sperm donations, or males who are coming in with a female partner, so the couple's being asked, coming in with a partner. The male may not be providing the sperm for the IVF. The sperm may be coming from an anonymous donor. So the individuals or couples who are seeking out IVF, and they're asked then, what is their preference? Okay, you're going to have a child by IVF. Do you want to know who the donor is or not? Okay, so sperm donation and IVF in particular are largely unregulated technologies in the U.S. And this is a big area of concern for a lot of people, particularly who do this for a living all the time. And we're going to spend a good bit of time today looking at some of the genetic implications. I want to give you an example. Good friend and colleague, Susan Rothman, is at the Cleveland Clinic, and she runs a company called Infertility Solutions Incorporated. They're one of the big IVF type of groups. What they really do is they work with IVF labs all around the world to train IVF technicians to identify viable sperm for in vitro fertilization. Okay? Now, when I say identify viable sperm, the existing technologies right now are largely based on what we call morphology or structure. How do the sperm look under a microscope? Okay? Many of the biochemical and genetic tests that we'll talk a little bit about today, you can do on sperm, but you can't then take that sperm and use it for in vitro fertilization because the tests destroy or disrupt the sperm. So you can look at sperm from a male and say, well, there's genetic abnormalities in these sperm, but you then can't sort of erase the test you've done in that sperm and use it in a fertilization event. So you're primarily basing this on structural defects. There are a couple different criteria. There's a World Health Organization set of criteria where there's a series of different factors that you look for. Do the sperm have a head? Well, if they don't, they're not going to help you out too much. Do they have a tail? If they have a tail, is it bent? Is it kinked? Is it the right length? Okay, are there abnormalities to the tail? You look at structures to the head that are involved in sperm egg binding and penetration. So there's a checklist, if you will, okay, that's imperfect at best. There's another set of criteria developed by a long-standing scientist in this field that many other countries use. So there's not even debate within the industry as to what criteria you use to determine what is a quote-unquote normal sperm. Many industries over the years have developed what are called computer-assisted sperm analyzers. So they're an attempt to computer automate the process, and they're okay to a certain level, but they're not perfect either. So by and large, we rely on human interpretation. So you see a panel like this, okay, and this is just a small subset. You see a bunch of sperm under the microscope. This would be a view that a technician would be looking at when they're going to select sperm for in vitro fertilization. Granted, in a projection like this is not the perfect resolution. It's not the same way you're going to see it under a microscope. But this is what the technician is looking at, okay? For about 8 or 9 years, I ran a group through the American Society of Andrology with Susan as a lead in this, where we went around the country and tried to teach labs how to analyze sperm in a better way, okay? And the reason why I point that out is that one of the first things we did was we ran workshops where we said the only people who can come to these workshops must be people who've been involved in IVF for a minimum of 10 years. So technicians who do this every day for a living. Okay, they look at thousands and thousands of sperm samples every day. Minimum of 10 years experience, you're going to come to a workshop. We'd show them this image. We'd say, how long would it take you to make a diagnosis on a typical image? And they'd tell us however many minutes. Okay, you have so many minutes. Make a determination here of the percent normal sperm and the percent defective sperm based on the criteria that you've been using. Okay, they would do that. We'd then show them this image. Do the same thing. Okay, now I gave you a pretty simple image here because if you look closely, there are some characteristics that will give this away. But this is the same image you saw in the previous slide, okay, just flipped around. And what they do is they show us this. We'd go back to this image, and they'd say, well, this image has about 20% defective sperm. or 30% defective sperm. And then they look at this one, they go, oh, this one's fine. 1% defective, 2% defective. And then when we show them that these were the exact same images, just flipped over, they couldn't believe it. Okay, and these were people who were doing this for 10 plus years, day in, day out. Okay, and you can imagine what the technicians were doing who hadn't been doing this very much or were very inexperienced at doing this. Complete lack of quality control in the industry, okay, and just perception of how you turn a sperm over. And when you view it like this, to give you an idea, this is a normal spermatozoan right here, okay? You can see some others that have some kink tails and other things. These are sometimes artifacts of the preparation. It doesn't necessarily mean they even came out of the male that way. Here's one where the head is abnormally large, which represents some developmental defects and so forth. It's a very tough call, even for people who've been doing this a long time. Now, that's just at the sperm structural level. It doesn't tell us what's going on genetically inside that cell. Okay, so this is a huge concern, a huge problem. I can tell you on the regulatory side, as we talk about some of these issues, there was a panel convened in Washington some time ago, and myself and Susan, a lot of other people sort of in and around this area were asked to come down and talk a bit to Congress about these sorts of issues and the concerns about these issues. And besides the fact that you're talking to people who largely don't understand science, let alone understanding evolution, okay, the philosophy, like, is so prevalent right now, is let's just kick this can down the road a little bit because you're only 2 generations into IVF. Well, I'm going to show you right as we walk through this little bit that from a scientific standpoint, even 2 generations into IVF, we can make some pretty good predictions as to where things are going. But they don't want to address that issue. Let's push it down the road. Now, the reason why I say that we can start to predict where things are going, and you're going to start to see sort of a turn in the discussion here as we look at the impacts on evolution, is because of another assisted reproductive technology that is getting a lot of popular use these days, it seems, called ICSI. How many people here know about ICSI or have heard of ICSI? Anybody heard of ICSI? A couple people. Anybody know what ICSI is? What does it mean or what does it stand for? Yeah. Yeah, absolutely. So it's what we call intracytoplasmic sperm injection, ICSI, intracytoplasmic sperm injection. You can see here came about as a practice about 14 years or so after IVF. So the basic approach here is this structure you see here is what we call a holding pipette. So it produces just a very light suction, so it holds the egg. Okay, so this is in diagrammatic form. Here it is in real form, just flipped around. Here's the holding pipette. Here's a human egg held to this pipette. And then you come in with an injection pipette. And it's a little tough to make it out, but this is actually the head of a sperm cell here and its tail here. And the sperm is injected right into the egg, directly into the egg. Okay. Now, if you follow me before on my real quick explanation of in vitro fertilization, we said sperm and egg are harvested, put together in a dish, and the sperm then penetrate and fertilize the egg. Here, we inject the sperm right into the cytoplasm. You put it right smack dab in the middle of the egg, okay? Now, why would you do this? Well, ICSI has been widely used for individuals with very severe fertility issues, especially male factor fertility issues. So the egg is in pretty good shape, okay? But you have very severe male infertility issues. Those kinds of issues where the sperm will not recognize and penetrate an egg in a dish by traditional in vitro fertilization. Okay, so you jettison it, you push it into the egg as a way to achieve fertility. You still let this grow in the lab for a few days, and then you implant it, and from there on out, it looks very much like traditional IVF. Okay, well, one of my colleagues who served on this panel when we went down to Washington, Dory Lamb, runs a major group at Baylor that is very much concerned about what happens when we start doing ICSI. Okay, and if you're really interested, there's a lot of really good literature. This was one of the first ones after ICSI had been out there for a little while that started to address some of the risks. And she was really worried about the risk because in the area of Baylor where ICSI is being used a lot, one of the first things they were starting to see were babies born with very low birth weights. And then developmental issues related to some of those babies, although not all of them, okay? Now, one of the many issues of concern in ICSI is a genetic condition that we call aneuploidy, okay? And aneuploidy is abnormal numbers of chromosomes. So on the left-hand side, this is a colorized image. You likely have seen what we call karyotypes before. This is a colorized image of a karyotype, where these are pairs of 23 chromosomes. And in this particular case, coming from a female, you see 2 copies of the X chromosomes. This is a nice, normal genetic karyotype. 2 copies of chromosome 1, 2 copies of chromosome 2, etc. 23 pairs of chromosomes, okay? This is a karyotype, a representative type of karyotype from some of the things that are being found in ICSI, okay? And I don't want to, again, I'm not showing you this to push the panic button or to make light of this, but in this case, this was taken from skin cells. These are also taken from skin cells. And what you'll see here is if you look, for instance, at chromosome 1, it's colorized in yellow, there's 3 copies here, okay? 4 or more copies of chromosome 2. That represents a piece of chromosome 1 that is on chromosome 2. Shouldn't be there, okay? And there's a piece of this chromosome missing. You can continue on through this scheme and you'll see increased numbers of chromosomes in many cases. You'll see these swaps, which we technically call translocations, where one piece of a chromosome is tagged onto another, okay? Multiple copies of other chromosomes here along the way. Aneuploidy, abnormalities in chromosome numbers, okay? Question? Great question. You can, okay, and we're going to look at that in a second. But this, as you see it right here, would not reveal itself necessarily early on. Can, but not necessarily, okay? So this is starting to show up. Now, this type of karyotype, okay, was from an individual who was asymptomatic at the time that the karyotype was taken. All right, and so in many of these cases, these kinds of chromosome abnormalities, what they will manifest in the future, one simply doesn't know, okay, because a lot of times the phenotype of the appearance won't necessarily be obvious, okay. So I couldn't even begin to tell you, we can't even get a handle on what percentage of individuals from ICSI have these kinds of abnormalities, because if you're asymptomatic, you're not going to be tested for something like this, okay. And then if you're tested 20 years later and you have some type of genetic symptom, you won't know necessarily cause and effect until you start looking at large numbers or cohorts of these kinds of individuals, okay? A recent study, which was the biggest done to date, involved 27,000, I'll come to your question one minute, please, sir. 27,000 children born from IVF, matched up against a very large cohort pool. Many millions of non-IVF individuals showed about a 42% increase in cancer within the IVF group. That was both in vitro fertilization and ICSI, okay? So the statistical predictions were that this is a greater frequency, much greater likelihood than in the generalized population. And that's a study that has gotten a lot of attention. It was one of the first big cohort studies of its type. We have a question here, Jeff. Yeah. Mm-hmm. Yeah. So good question. So this one was done back in. You can see here it was actually done in late 2003, 2004. It was published in a set of work in 2005. Okay. This particular individual from which the sample was taken has, among a number of different problems, developmental delays, cognitive developmental delays. And right now, the physicians who deal with this individual are very concerned about skin cancer, because the person is developing melanoma-like lesions, okay, pretty frequently, okay. And there's probably a lot of other, given what we know about the human genome and what genes are on some of these chromosomes, other delays that the sort of patient confidentiality, privacy issues have not made this particular individual, full health situation very well known, except that the parents have used this individual, as many others have, to raise warning signs. Okay. Question? Yeah. Great question. I'll ask you, hold on to that idea. I'm going to show you a couple things, and if I don't answer that question, let's come back and we're going to talk about that. Okay, so the question was this due to abnormalities in the sperm or the egg cells, or something in the process, the physical process of going through ICSI or IVF? Good question. Okay, so what I'd want you to think about in this context of the 204th birthday, if I did my math right, of Darwin. I wasn't sure if you guys started this by singing happy birthday or how things kicked off today. okay, is let's think about some of what I presented for you this morning with respect to IVF and ICSI, and sort of challenge the question that some people have posed and many people are changing their mind about, and that is, if these practices of assisted reproductive technology are pretty rare, you might imagine then they'll have relatively little impact on human evolution because you're dealing with relatively small numbers. Okay, but we already know many examples where the numbers are growing and people are very actively looking at this. Scandinavian countries, okay, tend to be one source of good information about where we may be going with this. And keep in mind, again, we're talking about a very, very short evolutionary timeline, right? 78 till now. Okay, so barely 2 generations, with the exception of people having children who are very young, barely 2 generations into this. Okay, on a natural selection timescale, minimal. Okay, but in Scandinavian countries where this has been looked at, in 2003, as many as 3% of the total births, okay, were born by ICSI, conceived by ICSI. It's an area that uses ICSI quite frequently. Okay, 1.7% of children in France conceived by ICSI. I meant to give you an actual number here. I didn't. ICSI, 300,000 children in the U.S. in 2003. Okay, so the numbers are not tiny. And when you're in countries where there isn't a lot of heterogeneity in the population, there's cause for concern. In Scandinavia, okay, is a group where we look at single gene genetic disorders like cystic fibrosis, which you likely are familiar with, in the general population, you can see what the incidence is. Now, the reason why geneticists are so interested in looking at things like this in the context of Scandinavia is if you have a generalized population where the incidence is about 1 in 2,200 to 1 in 4,500, that's live births that survive, okay? Then what you can do is take this kind of data and overlay it with individuals born to ICSI and IVF and ask the question, does the incidence of conditions like cystic fibrosis go up? And the answer in a very short period of time this has been done is yes, and dramatically so. Okay, 15 to 18-fold increase. Because one of the things that we know from these kind of procedures, when I walk you through this a little bit, okay, is that what you're really doing with ICSI in particular, but also IVF, is you're propagating infertility. Okay, now I say that in the context of being someone who's interested in using reproductive technologies to help couples have children, okay? But one of the things we've learned after multiple generations of this is that what you see in the sperm donor samples and the egg donor samples that represented infertility problems in the first place, those are being pushed across generation. So when you start to look at individuals that are a generation or 2 generations removed from the original cells that started this whole process, that they have many of the same infertility issues at the cell and molecular level that the donor cells did. So what you're doing is, from a selective standpoint, you're already now enabling fertility here, but you're propagating and you're in fact expanding and amplifying that infertility. Because normally if those individuals weren't having children, okay, you'd weed out, you'd have a selective pressure against those infertility issues. So many people are very worried about a quote-unquote infertility time bomb among children that come from assisted reproductive technologies, okay, because you're just propagating this issue. So why are you doing that, okay? I thought this group would be interested, so I know this is a thinkers group, about thinking about some of those Darwinian natural selective pressures. We talk about them as hurdles in the lab. So you have sperm samples that are isolated in the lab. You already heard a little bit about how inefficient that is as a process, and you're going to somehow get those sperm to mate with an egg, and you're going to make an embryo. Well, biologically, there are hurdles that those sperm have to jump through. And remember, I'm just keeping this on the male factor side. We haven't even really explored the female factor issues here. Okay, well, what are those? If we just look at them on the sperm side, so here we start with the sperm sample down here. Okay, if we were looking at sperm number, sperm concentrate, so someone walks in, they want to make their $150 that day. Okay, they saw the poster out in the streets of Red Bank. They want to make the $150 that day. What do they have to have in terms of sperm number, concentrate, to be considered a viable sperm donor? Anybody want to throw some numbers out there? We saw where that went before, but I'm taking bids. Give me some numbers. Give it to me in number of sperm per milliliter of semen. That's how it's done. 100,000 per milliliter. 50,000 per milliliter. Nothing like talking about semen concentration on a Sunday morning. Another number. Pretty good. What was that? Well, they certainly could fit on the head of a pin. So we actually did pretty well there. We're in the range. So 50 to 150 sperm per milliliter is considered the typical range for a fertile male. Okay? 50 to 150 million sperm per milliliter. Okay? Now, only a very small percentage of those sperm will actually be normal. So what people don't even realize, even in a fertile male, okay, having 50% of the sperm be normal is actually pretty high. Okay? So right away, when I showed you those previous photos and clinicians start telling us that only 10% are abnormal, it's nonsense, because even in a fully healthy male, only about 50% of the sperm are really viable sperm. Okay? Now, the reason why I say that is because if a man has less than 50 million sperm per milliliter, so you come in the lab, I have 40 million sperm per milliliter, a person's feeling pretty good about themselves, they're considered clinically infertile. Okay? And what that tells you is, given the sheer numbers of sperm we're talking about, even having many millions of sperm per milliliter, is that there are so many barriers to fertilization as an event that your likelihood of conceiving is very low, even if you have many, many millions of sperm per milliliter. Question about Jane? Yeah, good. In terms of the sort of the age limit or cutoffs, is that what you're thinking about? Yeah. So it's interesting. Most clinics like to see healthy males come in somewhere between 18 and about 35 years of age, 32 to 35 years of age. But that can vary a little bit. I mean, I certainly know some clinics that use a cutoff of 22, 25. Okay. Now, that's just for soliciting anonymous sperm donors. Obviously, if a couple came in and wanted to do IVF or ICSI, and either one of the partners were older, okay, I mean, they can certainly do that. I mean, if an individual came in, was in his 60s and wanted to donate sperm for use by his partner, you can do that. Okay. All right. So I want you to think about the numbers issue that I was telling you. So the number of sperm and concentration is a big deal. Okay, because even if you have 25 million sperm per milliliter, your odds of natural conception are very low. You're considered clinically infertile. Okay, because of these barriers we're going to talk about. I mentioned very briefly this idea of all kinds of sperm developmental defects. These are the things we can see to the naked eye. These are some of the easier ones to pick out. Even those we can't do very well. We can't do this very well in the sense that we can look at chromosome number defects. We can find them, okay? But again, keep in mind that if you find a sperm and you say its chromosomes are normal, the tests you have to use to do that prohibit you from using that sperm for fertilization, okay? Another barrier. These are natural biological barriers that are happening, okay? Sperm transit. So even if you have 50 to 150 million sperm per mil, eventually only very few are going to get to the egg, right? Because one of the first things they have to do is they have to make their way through the male reproductive tract. They don't actually swim, but they're pushed in various ways. But when they get in the female tract, they have to swim. So right away, if you don't have a tail, or your tail isn't producing sufficient metabolic energy, you're not making it to the egg, right? And some large percentage of sperm never make it there because they just can't swim. They don't have the motility, okay? And there are some diagnostic tests where you can measure motility in the lab. When sperm get to the egg, they get pretty excited. Okay, there's an event called hyperactivation, and they look like kids in a candy store or kids on Christmas Eve. They start twitching, they start shaking, the tails start moving very quickly, and metabolically they're preparing for fertilization. I won't bother you with all those details, but they undergo a series of enzymatic changes on the surface called capacitation. In other words, a series of enzymatic changes that now expose certain proteins on the surface that make them eligible or capable to bind to the egg. Okay, and they sequester those proteins while they're in the reproductive tract because you don't need them. Okay, but they expose those sites at the time of fertilization. And again, only a small percentage of sperm do that. Now, on top of all those things, okay, if a sperm does bind to the egg, I probably should just move this one up a little, but here you see an egg and it's coated in sperm. So many will get there, okay? Many will get there and start to bind to the egg. But the one that makes its way through a certain critical layer of the sperm elicits a chain reaction that's called the block to polyspermy. So for those of you who've had biology before, I may be bringing back good or bad memories when I talk about stuff like this. But in the block to polyspermy, what happens is when the sperm penetrates and hits a certain part of the egg membrane, there's a wave of calcium that's propagated across the egg. And it hardens the surface of the egg effectively at the biochemical level so that other sperm cannot penetrate. Okay? And so you were the winner. You got there first. You block everybody else out. And that's to prevent multiple sperm from penetrating that egg. Okay? Now, if you see where I'm going with this, we're bypassing all these barriers right away. Okay? Whether it's in vitro fertilization or ICSI. Okay? For ICSI, we bypassed all this because we went right inside the egg. We bypassed all of this, with the exception of this piece. Sperm egg binding, there's a coat on the egg called the zona pellucida. It's a sugar-like layer that the sperm have to bind to. Well, when IVF is done, you actually remove that layer from an egg. You peel that off because you want to make it easy for the sperm to get in. Well, that layer is there to ensure that only sperm with certain proteins that can bind to the zona pellucida can make it. And now you just say, well, let's let everybody in, open admissions. Okay, you don't have to have those proteins on the sperm. Okay, so you've stripped away a barrier. Well, in ICSI, you go right into the cytoplasm, right? So you don't have these barriers. Sperm egg fusion, even if a sperm penetrates the egg, there are fusion events that have to take place to eventually create that block to polyspermy. Okay, and then inside the cell, there's what we call the mixing of the pronuclei, meaning the nucleus from the sperm, the DNA from the sperm, and the nucleus from the egg have to mix. And it's at that point, biologically, we say there's conception because the two genomes have mixed now. Okay, well, this oftentimes doesn't happen in ICSI because you force-fed a nucleus into the cell. Okay, and then you have implantation. Okay, which is an issue, obviously, naturally, or whether it's in ICSI or IVF, and a whole lot of issues around infertility here. You likely perhaps may know people who can get pregnant but cannot maintain the pregnancy for various reasons, okay, with respect to the placental environments. That's a whole nother sort of discussion there. Okay, you get the take-home point. A lot of barriers, a lot of hurdles, all right? And we jump over most of those when we do IVF, and particularly when we do ICSI. Now, because of a lot of these kinds of issues, over this period of time as these technologies have developed, many people have started to ask a variety of ethical questions about what we're doing with IVF and with ICSI, okay? And I mention this and use this quote as an example because what I want to show you is that as these assisted reproductive technologies have moved along now, things like the Human Genome Project and genetic engineering and genetic testing have enabled us to be able to ask very different questions now about the ICSI and IVF world. And that's kind of where I want to focus the latter part of this talk. Fairly early on, okay, going on now for, well, almost about 15 years, but really accelerating in the last 6 years, is the ability to do what's called PreImplantation genetic diagnosis, okay, or PGD. How many folks have heard of PGD? When you look in the newspaper, on TV, when companies advertise IVF, if they do PGD, they'll usually be big, bold letters, we do PGD, because that's a big deal now, okay? Well, just let the words do the work for you. PreImplantation genetic diagnosis. You've made an embryo in the lab, and now you can do some type of selection or screening of that embryo and decide whether to implant that embryo, okay? So here's the way it goes. Now, the first application of PGD was embryo selection for gender because it was really easy, okay? There were other techniques to do gender selection. It turns out that if you know your genetics pretty well, you know that the male Y chromosome is pretty small and the X chromosome is big. And it actually turns out that Y-containing sperm, okay, are therefore lighter at the molecular level than X-containing sperm. So you can do a very simple technique in the lab where you spin the cells and you can separate them out by their density, by their weight. But that was effective to about 80%. But farmers have been doing that for a long time. You can also let the sperm swim up a straw, okay? So the X sperm are a little bit slower than the Y sperm because they're lighter. All right, so you can let them swim up a straw through a very thick sucrose solution or sugar solution, and with about 75% efficiency, you'll get males at one end and female sperm at the other end. Okay, again, in the dairy industry, people have been doing this for a long time. Okay, all right, so that was one technique that was happening in IVF early on, but with pre-implantation genetic diagnosis, you could pinpoint with 100% effectiveness gender selection and a lot of other things too. And here's how it works. Here's that holding pipette I showed you before. Okay, same sort of thing. Provides just a very light suction. There's a suction pressure going this way. Now, this is an embryo that is at, this is at the 8-cell stage. So typically PGD is done between 8 cells and 16 cells. So a sperm and an egg now have fused, regardless of whether it was IVF or ICSI, and it goes 2 cells, 4 cells, 8 cells, 16 cells, okay, before it gets implanted. As early as the 8-cell stage, what you can do is come in with another suction pipette, and you can suck away one of those 8 cells. So you see the cell going in here? Okay, so this embryo now is left with 7 cells. There's some risk there. It sometimes will kill the embryo, but generally speaking, the embryo is not going to miss that cell, okay, because at this point, all the cells are undifferentiated. They all are the same. None of them have decided yet whether they're going to be a skin cell, muscle cell, bone cell, whatever. That comes later, okay. Less risky if you do it at the 16-cell stage. You can now take that cell and you can subject that cell to genetic analysis, okay? And I'll give you a sample of what kinds of things you can analyze it for. But one of the first things you can do is you can do a karyotype like you saw many slides back, the colorized karyotype, and you can look at its chromosome number. So that's where you can see right away if it has an X or a Y. So if it has a Y and you're a couple who wants a boy, you can use that embryo, okay? If it has 2 X's, you want a female, you can use that embryo. So sex selection is really routine, very easy, okay? We do this in the lab all the time, not with human embryos, because we don't do this as routine experimentation with human embryos, but we do this with some of the organisms we work with in the lab when we study reproduction. We do this particularly with rats and mice. All right, so sex selection happened early on, and it was pretty routine. And then what started to happen shortly after that was, because of the Human Genome Project and our understanding of a lot of genes, you started to screen for lots of other things. So this is just a simple little cartoon here where how to increase the chance of having a green-eyed, blonde-haired daughter, okay? Because we know what those genes are now, and the same cell selection that we did in the previous slide, instead of testing for gender now, you test it for eye color, or you test it for hair color, okay? And then you make a decision to implant or not, okay? I'm going to show you more on this. Back in the early 1990s, Glenn McGee, who was a bioethicist at that time at the University of Pennsylvania, wrote a provocative, popular book about the concept of making designer babies and this idea of a perfect baby. And it was meant to be provocative and floating a lot of ideas out there. And if you're interested in that, I would encourage you to go and take a look at that book. But it was really spot on in some ways, because we've been doing, in that sense, designer babies already for quite a while here. So using the example I just showed you of PGD, what kinds of things are being screened for? Well, we talked about the sex piece. That's been pretty routine. But what initially started happening by PGD is a major, major focus on the single gene disorders, because these are the ones that were the easiest to test for at that time. Okay, so for example, the common ones that you may be more familiar with, cystic fibrosis, sickle cell anemia, albinism, Huntington disease, because there's a single gene. You can analyze that gene through a variety of tests and know if you have that gene, you will develop this particular condition, or this embryo may be a carrier for that condition, okay? So immediately, PGD was being used to screen and select against these kinds of conditions, okay? Abnormalities in chromosome number, the aneuploidy kinds of conditions. One of the most well-known examples that you may be familiar with is trisomy 21 or Down syndrome. The same technique that I just showed you on PGD, you use to look for chromosome number, and then would make a decision whether to terminate the pregnancy or not. Very dangerous, slippery slope kinds of things, okay? Now, you add on top of that a layer where now increasingly, as we get to understand the human genome, okay, that these kinds of screens are being done for genes involved in Parkinson's and Alzheimer's. We can go on and on and on. Eye, skin, hair color, height, even weight, or speed, okay, which is a really loose one, because there are genes that are potentially implicated in making one fast twitch fibers in their muscles or not, but it doesn't mean that that's the sole factor in determining whether someone is speed. We've left all of the behavioral and environmental issues out of this conversation. I'll come to your question in 1 minute, please, sir. And what's even more scary is where this is going in the future, because there are a lot of behavioral genes being explored, okay? where the prediction of these behavioral genes is only very loosely associated, if at all, with behavioral conditions, okay? Think about intelligence for a second. I joke with my students in genetics, there's not one intelligence gene where you either have it in your Einstein or you don't and you're an idiot, right? There's a lot of shades. There's a range, okay? There are genes that play a role in cognitive development, in neurobiology, in brain development, and so forth. But we know really very little at all about the interplay of those genes, about the effect of environment, and all those other factors, right? But yet, from a genetic screen standpoint, you can tell if you have the gene or not, okay? Whatever the gene is. Same thing for a variety of other conditions. And incidentally, who's getting a lot of attention down in Washington for the last 10 years or so are the insurance companies who want access to not only genetic information like this from PGD, but also you can do these tests on adults now, of course. And in terms of raising insurance rates, just like you would for a driver or a smoker, they want to do the same thing on genetic predictions. And so far, there are a number of genetic privacy acts that have kept that at bay. But that'll be very interesting. And where I want to go to finish up this morning is to show you that the field is moving towards what are being called destiny tests now, okay? If you follow a little bit about the human genome, we are in the very near future here going to get to the point where an individual genome, all the DNA in a person's cells, okay, full set of 23 chromosomes, can be analyzed for about $1,000. Right now, when the Human Genome Project started in the 90s, it was a 15-year effort at a proposed $3 billion price tag. And like any other technology now, it's coming way down. Individual genomes are being done in about 2 weeks now, okay, and the cost is under $5,000. So the $1,000 genome is coming, okay? It's going to be there, okay, which is another unregulated issue. But the concept now is not only can you test for individual genes, but you can look at the genome on a large scale. All roughly 20,000 human genes, all 3 billion base pairs, and you can do it in a PGD type of test, okay? Now, I mention this because some of this is already happening. In the United States, of 190 clinics that do PGD, okay, 3% of these offer the services to intentionally select for an individual with a genetic disorder. This is a highly ethically provocative issue. In other words, if you're a couple and you go in and you want to have a child with a genetic disorder, they will help you select for that child. So you may have heard of some well-publicized cases of a couple that wanted a child with dwarfism, okay, and wanted a child that was deaf, and they were granted those wishes, okay. So this is not selecting against some of those conditions, whether you believe that should or shouldn't be done, but selecting for those conditions, okay. Good question. Why would they? But to be like themselves is one reason. Other thoughts there? I love the passion. They're nuts. These are exactly some of the kinds of emotions that this sort of topic evokes. Yep. That's another interesting one. I don't know that I've actually heard that one before. Maybe they need the money and they can get some kind of aid from the government to support a child with one of these conditions. Let me show you another one or two, and we'll wrap up and then have some ongoing discussion here. What's intriguing about these destiny tests now is they're not just for the assisted reproductive technologies anymore now, the PGD that I showed you. A big move now is in what's called in utero testing. About 2 years ago, a group very successfully demonstrated that when a woman is pregnant, about 3%, 5% of the DNA circulating in her bloodstream at a certain point in the pregnancy is from the developing fetus. Okay, and now with molecular techniques the way they are, you can go in a blood sample, so a non-invasive way, take a little bit of mom's blood, and you can identify the DNA from the fetus in mom's blood. Okay, so this is in utero testing, so mom is carrying a baby now at this point. Okay, so what you're looking at here, this is just a very simple example. This is a segment of chromosome, so let's just say for sake of discussion This is DNA from mom for chromosome 1. This is DNA from dad from chromosome 1. The fetus, okay, will be a combination of these DNA samples, and there's very relatively easy ways in which you can identify that, and you can pick that out of the maternal bloodstream, okay? Oversimplifying it, but you can do that now, okay? So you will know in utero, okay, some of the results of some of these kinds of tests, okay? Let alone just preconception testing. Preconception testing now is that you can test sperm and egg cells. You still can't use those sperm or egg cells for conception. But what you now start to do is a very slippery slope of making predictions. This is a company, GenePeaks, was developed by Lee Silver. Lee was a prominent geneticist at Princeton University for a long time. He wrote a book I'll show you about in a minute. I cut some of my teeth in the world of genetics in Lee Silver's lab. And what Lee put together now is a company that will sequence a woman's genome, sequence a man's genome, and digitally, look at this concept, digitally weaving together the DNA of the prospective parents. So this is done with a sperm and an egg cell. Okay, so he takes multiple sperm from the male, takes a couple of eggs from the female, sequences their genome, and now you have this set of genes here, this set of genes here, and digitally overlays them. This is using computers, and tries to make predictions about genetic compatibility. And what spits out of that is you have genes that match up that increase your likelihood or probability for X. Or you have genes that match up and increase your likelihood for health in this area. All predictive stuff, okay, of which many question marks reside about how accurate any of this is, okay. Doesn't allow you to go and select the best sperm and egg cell that are the best digital match, but is genetic digital matchmaking. It gives couples the opportunity to decide whether they're genetically compatible, okay, at the computer science level, and then to decide whether they want to roll the dice in the genetic risk, okay. Very, very scary concept. Also moving in the direction now where in the last year and a half, 2 years now, growing sperm and egg cells in a dish is happening. This is from stem cells, okay? And that's a topic for a whole nother day. But the point is now, if you can make sperm and egg cells in a dish, you can also genetically manipulate them in the dish. So you can introduce into them genes that you want. And we've been doing this in the lab for a long time. So we can actually take cells in the lab, and if I want to test out whether a gene I study protects mice against a yeast infection, I could introduce that gene into cells and make mice that have a lot of that gene. They overexpress that gene, okay? We didn't have the ability until recently to make sperm or egg cells from humans now, and now that that possibility is there, you can make genetic manipulations, add genes. Can't remove genes too easily, but you can add them, okay? So now you add a whole nother layer of the ability to design a baby, okay? If some of the things I've mentioned to you today you find of interest, there's a lot of great popular reading. I mentioned Glenn McGee's book out there. This is actually Lee Silver's book from some time ago called Remaking Eden. And recently, Max Mellman, and this is last year, 2012, Max Mellman is at Johns Hopkins, wrote a very provocative book on dystopian nightmares and transhumanist approaches which deal with the issue of designer babies and all these implications of a lot of the kinds of things that I've been talking to you about this morning. Okay, with that, I will thank you for your attention and hopefully leave you with some things that you're thinking about or intrigued by. Question? Yep.
All right, hold on. We're gonna do questions and stuff. I'll hand out the microphone and we'll go one by one. So raise your hand. I'll try and keep the order that you want to ask questions in, just to keep this orderly.
One of the big questions for a group like this we've not really gotten into, but with all we know about molecular biology today and reproduction, why do most religions still believe in virgin birth?
Yeah, I'm afraid I'm going to leave that one unanswered for you today. But maybe my colleague Stan Green here has some people in the philosophy and religion world who can come in and talk a bit more accurately about that. Yeah.
Hi, I've read about couples on a first or second date where one asks the other for their credit score. how many years away are we till one asks the other for their genetic profile?
I'll tell you, you're spot on. So there's a cartoon slide that I use in one of my courses where there's two people at the bar, and it's basically there's the bubble conversation is, you know, so what's your Alzheimer 3 gene scale? This is really something that we're talking about right now. In fact, there are companies out there, talk about an unregulated industry, there's a whole cottage industry of companies that will do genetic tests for people now, what we call direct-to-consumer genetic tests, the DTCs. Okay, if you go out and do a Google search and look for a company called 23andMe, by no means an endorsement, but they're getting a lot of attention. 23andMe, you send them cheek cell or hair cell sample and plunk down $100 or more, depending on how many tests you want done, and within a couple weeks you get a test back. Now I want you to think about this. You're sitting in your kitchen, okay, and you get an envelope coming from 23andMe, and there's a whole bunch of genetic tests that have been done to your sample, okay? And you get some indication. You have this gene, you have that gene, your percent or your probability of this condition is X. You're sitting there by yourself. You don't even know what the tests mean, okay, necessarily. You don't have a genetic counselor with you if you really wanted to talk through and deal with some of those issues. And this industry, okay, is entirely unregulated in the sense that there's no quality control for false positives, false negatives. Most of these companies don't have to reveal to you how many times they do the tests. A lot of times they do it once, okay, one run. And I'll show you, my students will tell you in the lab, there's a lot of things they do once and will never happen again, never work the same way again. So they're not required to do it X number of times and validate their results. These tests are extraordinarily sensitive based on a technique that amplifies DNA, much like what happens in forensics labs. And so the cross-contamination between samples is extremely problematic if you don't work in safe or sterile lab environments. So because that's unregulated, then no one even knows the relative percentages of false positives and false negatives. Some groups have been doing some sleuthing to deal with this issue. They've been sending multiple samples of the same material under different names, and they're getting some very interesting different results back, okay? So unregulated industry, direct-to-consumer testing, and you already have that in your back pocket now. Okay, and so it's not that far-fetched that conceptually one would be able to do that. I mean, this notion of a number of years ago when this idea of $1,000 genome came out, it was issued as a challenge. So this group issued $10 million to the first company that would come up with $1,000 genome, and everybody said, "Oh, only a few companies will enter because it's going to be impossible to do." And now it's just a question of which company is going to get there first, because there's 20 companies lined up and they're all very close, and one company's already saying now we're going to have a $500 genome available relatively soon. So it's going to happen. Okay, it's going to happen, and if you want to do it, you can plunk down your money and have it done. Insurance companies aren't paying for a lot of that kind of stuff. Sometimes they pay for PGD if there's a family history there and a good reason to have it done. But the scenario you're talking about, Robert, is not far away. Yep.
Okay, one more thing is now you're getting back to that. When you were mentioning Alzheimer's and Parkinson's and exposing the potential risks of having it, when they do these tests, how much accountability is there for environmental factors during the life of that individual, whether they're going to have that Alzheimer gene show up, because like different environmental factors might cause genetic mutations during the life of the individual?
Yeah, absolutely, no question. A great point, and virtually no consideration, because no one has a way to be able to do that. Right, so it's presence or absence of a particular gene. I mean, I'll give you another example that's very controversial. You may have heard that in conditions like Alzheimer's, for example, you see these things called neurofibrillary tangles. There are plaques. So you see a deposition of protein material that shows up when the brain is imaged, okay? And for many, many years, people made the cause and effect. They said those plaques or tangles that show up, okay, are the cause of Alzheimer's. And we know what proteins are in those plaques, and we know what genes cause those proteins. Well, now what the field is really thinking about is that they're not the cause of Alzheimer's, that the proteins in that plaque, okay, are an effort to get rid of the dead cells in that area and regenerate the cells, and something else is causing the cells to die in the first place. So the Alzheimer's screening test has been for a protein called ApoE, a protein that's part of those plaques. But what that screen probably tells you is your ability to ward off or protect against plaque development. And there's a lot of evidence that if you have certain variants of that gene, that you would be protected to a certain level from Alzheimer's disease as opposed to getting it. Okay, so even at the molecular level, we don't know what a lot of these genes do really well. we know they could be associated and increase the likelihood of a condition, but we don't factor in the environmental causes in any real way. There's a lot of interesting twin cohort studies that are done in this area, of course, where you can take twins who have identical genomes, but they may be split early on in their life and may have grown up in different environments. And you can look at Alzheimer's and other conditions, alcoholism and other things. And we know quite clearly that environment has very significant impacts on the expression of those underlying genes. Okay. Other genes, not so much. I mean, other genes, if you have the gene for Huntington disease, for example, or if you have the gene for sickle cell anemia, then you know what the phenotype is going to be.
You mentioned that data was being collected on the next generation,
Yep.
or maybe the generation after that. Can you tell us more about what you're seeing generations on from these individuals?
Yeah. So part of what's interesting there now is there's about 6 or 7 what I would consider major global initiatives now to look at these kinds of issues in terms of cohorts of offspring. And I think when you start looking at 20,000, 30,000, 40,000, 60,000 individuals, you have some statistical validity to some of the kinds of things that we're looking at here. And there's no question that on the ICSI side and then the IVF side, that you see a much higher than normal incidence of a range of different genetic conditions being pushed down into those offspring. There's no question about that. Okay, now it's not 80 or 90 or 100%, but what I also say is you haven't tracked most of these individuals full term from birth to death either. Okay, so the challenge is you've got individuals at all different ages, so you're trying to do age match studies, and then you're trying to track this person. Did this person live out a healthy life? Did they live to 80-something years old, or did they die earlier? And if they died earlier, can we track that incidence to some of these assisted reproductive technologies? Most of those generations, those individuals have not lived out a full, what we might consider a full lifespan, so we still don't know the answers to those questions, even about longevity. Even a basic question, okay, is there a change in longevity of children born to IVF and ICSI? We don't know yet. But those are some of the kinds of things that are being looked at. And as I said, I don't mean to say that to sort of to push the panic button on this issue, except this is a genetic reality that is very clear, even just a few years into a lot of these kinds of studies. Did that help answer your question, Drew? There are some interesting issues around diabetes and obesity generally, and it has to do with this. Most people think it has to do with this low birth weight issue. One of the things that can happen in the children early on from a low birth weight standpoint is, for whatever the abnormalities are, metabolically they're not charged up to deliver proper nutrients to all the cells of the body. So what happens is you get a sequestration of nutrients. So for instance, sometimes you get overload to the liver or overload to the pancreas, okay, that creates organ-specific issues that manifest themselves as metabolic problems later on down the line. So there's low birth weight, but the nature of the body is to maximize resources to certain key organs. So you're driving nutrients away from the brain, for example, it causes developmental problems, then you wind up with liver or pancreatic overload that causes diabetes later. That's another type of example.
I've got an easy one for you. Boxers or briefs?
You left out a third possibility.
Commando.
You're right. Well, the temperature effect is a very clear effect. In fact, Scandinavia's been doing a lot of this because you look at men who go in these hot springs and pools and other things around various times of the year. It's where some of these first studies about the relationship between temperature and sperm fertility came about. So there's no question, just a couple % difference in the temperature of sperm development makes a big difference in terms of sperm number. There's a lot of, getting off this issue a little bit, there's a lot of very interesting studies right now that are looking at industrialized nations, and you may have heard a bit about this correlation between decreased fertility in industrialized nations and decreased sperm count. And so there's all kinds of speculation about environmental toxicants and other things of the industrialized age and their impact on sperm. Generally, from the big numbers of individuals that have been studied, it does look like globally sperm count is on the decline among men, but the cause and effects of and all those pieces of that are multifactorial and complicated to say the least. But yeah, briefs are commando. Boxers are commando.
Okay. Earlier I asked you about in ICSI sperms versus process. In more generally, I was surprised you mentioned that half the sperms are not viable, and predictions are being made about the outcome of fertilization. So when you are looking at prediction, presumably you are only looking at DNA from the eggs and the sperms. So are all the DNA from sperms identical? Obviously, as ages change, something changes because you prefer younger sperms.
That's right. So great question. So a couple pieces of that, and let me make sure I tie it all together there. First of all, some of it is certainly genetic issues of the sperm, okay, to begin with. So when you select sperm to use for a procedure like ICSI, okay, remember that each sperm not only has an X or a Y, but it has the other 22 chromosomes too, right? And so we still have this problem that if you look at sort of the Lee Silver approach, he can sequence those sperm and sequence a lot of them, but he can't sequence all the ones that a person makes. But he can sequence a lot of them and say, on average, X percentage of your sperm show these defects or don't show these defects. But you can't then take the best sperm and use them for fertilization. You can just do it by how they look, okay? But how they look only shows you a very small piece. ICSI as a procedure then also adds another layer of problems because you're taking sperm that in many cases would never even make it to the egg. I mean, ICSI is being done, for instance, on men who have sperm that don't swim at all. When I said severe fertility, man comes in, their sperm, they're just sitting there, they're dead. You dilute them, no matter what you give them, they don't move. Caffeine is a great one for you. What you do is when you have the sperm on a slide, they're immobilized in a very thick fluid so that they don't swim while they're in the male and use up all their energy. So what you do is you put them on a slide and you dilute them. And as soon as you dilute them, they start swimming. And you actually add a little bit of caffeine, and that gets them swimming. The ones that swim, you go after those. The ones that don't move, you leave them behind. Okay? Well, you can dump a cup of caffeine on some of these cells, and they won't move at all, but they'll still be used for ICSI. Okay, which is highly controversial. So you're taking sperm, there's clearly something wrong with them. And what we are finding from these kind of tests is that if they can't swim, then it's not just defects in the swim genes, okay? Because we always think about the X and Y chromosome, the sex chromosomes, as being only involved in sex. But what the Human Genome Project has told us is that many genes involved in sex are on the X and Y, but many genes involved in sex are on the other 22 chromosomes too, just like there are fertility genes on the other 22 chromosomes. So the X and Y determine maleness or femaleness, okay? But fertility is not just restricted to the sex chromosomes. So when you have a sperm that doesn't swim, okay, it's generally not just because its genes involved in swimming are defective. It's going to have other defects too. Okay, and so you're taking bad sperm in the first place. You're introducing them artificially into an egg. So you're stripping many of those barriers that are required for normal fertilization. Okay, so those two things together represent the real big problems. Is that fully? Yeah. Okay, let me talk some more.
I think we're just going to get two more questions here before we go.
Is there something, and supposedly in that situation, it's the egg and the sperm of the actual father and mother. So I have a couple questions. Are they counseled about all this? Are they counseled differently? And will the same things happen? And then what about their children? So.
All great questions. So typically, what the best clinics in the world are doing when a couple approaches that clinic to do IVF, first of all, you're going to query the patients, why are they there? Have you been trying to conceive? And you, as you know, being close to this, there may be a long history of attempting to conceive unsuccessfully, okay? And it could range, as I mentioned before, even from getting pregnant and then not carrying the pregnancy to we've been trying unprotected intercourse for a long time, and no matter what we do, we try hormonal treatments, we try timing ovulation, all these other things, we cannot conceive. So the first thing a good group is going to do is do that patient history, okay? And then they're going to even go back a generation or two. As best you know, on your mom's side, your dad's side, has this been an issue? And sometimes it is, and sometimes it's not. Okay? So you get that information collectively. As part of that panel, you need to know a bit about whether there's a prevalence or incidence of certain conditions in your family. You have a history of diabetes, heart disease, a range of different genetic conditions. That's all part of the information gathering, right? And then, of course, you have to really look at the biology of the male and female. You have to ask the question, is the woman making eggs? If she's making eggs, can they be fertilized? And there are ways you can assess that. As I describe all this, you see the costs are going higher and higher, right? You analyze the sperm. First of all, is the man even making sperm? Okay, a number of couples will say, oh, sure, I know I'm making sperm. I mean, without getting too graphic here, a man makes semen. It makes a fluid. But 90% of that fluid is not sperm. 90% of that fluid is coming from other accessory organs. So a lot of times people say, well, I have an ejaculate, I'm making semen, but there may not be any sperm in it. So again, first thing that's going to be done early on is that analysis has to be done. If there are sperm there, what percentage of them are normal? Do they swim? All those kinds of issues. Sometimes you see men, I just didn't really have time to work this in, but let me show you pictures where you can look at a semen sample where 3/4 of the sperm don't have any heads on them. They're all tails. And the head, of course, has the genetic material, right? The head has the chromosomes. Okay? So you have to start going through the biology there a little bit. Now let's just say at the end of the day that maybe the sperm aren't swimming or something like that, right? Now you're going to make a decision to do something like ICSI. Unless you really do a complex battery of genetic tests, you still don't know what underlying genetic conditions might be in the male cells or the female cells. You see what I mean there? So that therein lies the complexity. And you asked the question, are people made aware of this? In my experience, and I've seen family members and friends go through multiple rounds of IVF. I certainly don't encourage them to do ICSI, I can tell you that. But having sit in on some of those sort of patient types of sessions, I think the best groups spend a lot of time talking about the risks, and it's risk assessment, risk reward, right? Risk management. You want to have a child. Okay, here are the risks associated with having that child. You have to do the risk-benefit analysis.
Could you give me just a brief definition of epigenetics?
Oh, great.
or is that for another forum?
I'll do my best, but it's fascinating. Fascinating. Okay, so here's the idea. Bio 101, epi means around or near. Okay, epigenetics refers to changes in the genome that do not affect mutational conditions of DNA. So they don't affect an A, G, T, or C. So there's no mutation when we think about the bases, but they are changes in and around the DNA that affect how the DNA functions. And a lot of those changes have to do with how the DNA folds. It has to do with proteins that bind to the DNA and other chemical changes that happen to the DNA. There's a technical chemical change called methylation where you add certain components. So the difference of epigenetics is it's not a mutation. It's not a change, AGTC, like sickle cell is a mutational change. If we just sequenced your DNA would look just like somebody else's. But you might have diseases that the other person doesn't have because of these epigenetic changes that are happening around the DNA. Folding, as I mentioned, and other chemical modifications. Huge area of biology now because we know the environment affects epigenetics. And this is one of those areas I sort of subtly alluded to it before without saying epigenetic. These transgenerational changes I was describing to you, we know that epigenetic imprints last multiple generations. And that's really complex because it's easy to tell if a mutation's moved from generation to generation. You go to a certain place in the genome and you figure out, does this person have a G when they should have a T, right? But epigenetic changes now add another layer on top of this. And there's been, we know very clearly that epigenetic changes which are imprinted in an early embryo, the way DNA folds, the proteins that bind to DNA happen early in an embryo. We know that those changes can manifest many generations later. And when you do things like IVF and ICSI, okay, especially when you work with defective sperm, they are an epigenetic mess. And especially when you take sperm from an older male or even eggs from an older female. I mean, that's why, you know, eggs, as a woman gets older, they have more epigenetic issues, not only just mutational issues. Now you start an embryo with the epigenetic problems and they carry generation to generation. Last word on this, I know this wasn't a short answer to your question. Cloning as a practice, one of the biggest problems with cloning is that you start with cells that are older. So Dolly the sheep was started with mature cells from a sheep, mammary cells from a sheep. The epigenetic imprints in cells change as you age. So if you make an embryo from old cells, they're imprinted. The whole organism is imprinted to be old. So Dolly died of pneumonia-based complications, but showed all the signs of an accelerated aging because she was birthed from an old cell. Okay, so huge area. Does that give you a little bit better sense? Non-mutational changes, things that happen around the DNA, but it's not a mutation in the DNA.
All right, thank you so much. Give him a big round of applause.
Great. Thanks, Stephen.
Thank you, Michael.
Thanks for your attention. Appreciate it.
Thank you for listening to the Red Bank Humanists podcast. For more information, visit redbankhumanists.org or follow us on Twitter @RedBankHumanists.